Okay, let's unpack this. Enzymes. I know, it might sound like we're cracking open a long-forgotten biology textbook, but seriously, these molecules are the unsung heroes making life as we know it possible.
Absolutely. They're everywhere, doing
Think
everything.
about it from digesting your breakfast to the, well, electrical activity powering your thoughts. Enzymes are the engine. On this deep dive, we're plunging headfirst into the world of these incredible biological catalysts. Get ready to feel like you've unlocked a secret code of life itself.
And what's fascinating here is the sheer scope, really, of what enzymes accomplish. We're diving deep from their fundamental definition, first laid out by pioneers like James Sumner,
Right.
to the state-of-the-art techniques scientists use today to study them. We'll explore how enzymes are classified, the mechanisms that enable their biological magic, their roles in health and disease, and the cutting-edge tools that allow us to study them right down at the molecular level.
Exactly. We've got a seriously deep stack of material here, looking at the foundational principles and the advanced applications of enzymology.
It's a huge field.
Our goal is to equip you with a mastery of enzyme concepts, not just knowing what they are, but truly understanding how they function with such incredible precision. So settle in, because we're about to get intimately acquainted with the molecules that make life tick.
Okay, let's begin with the basics then. What exactly are enzymes? As James Sumner, a Nobel laureate, put it, an enzyme is a catalyst of high molecular weight and biological origin.
Sounds a bit technical.
It does, but the crucial part is catalyst. They dramatically speed up biological reactions, and those reactions, they're the foundation of life itself.
Right. Think about everything constantly happening inside you right now. Enzymes are the workhorses breaking down the food you eat.
Yep, releasing energy, getting building blocks.
Providing energy and the building blocks for everything else. Proteins, DNA, cell membranes. They're also the master builders, assembling those blocks into complex structures.
And they're involved in harnessing energy for all sorts of cellular processes, from muscle contraction.
Through the firing of neurons, allowing you to think and, well, follow this conversation right now.
Exactly. And while most enzymes are proteins, there are important exceptions. Ribosomal RNAs, for instance, key parts of the ribosome, the protein-making machinery.
Right.
They have catalytic activity. And then there are ribozymes, actual RNA molecules that act like enzymes, doing things like cutting or joining other RNA molecules.
We'll definitely circle back to those ribozymes later. Sounds fascinating.
Yeah, the whole RNA world idea.
So enzymes are essential for so many processes. What happens when things go wrong with them?
That's where the biomedical importance really comes in. Changes in enzyme quantity or their activity. Well, they can have direct and sometimes devastating consequences.
And those changes can come from anywhere.
Pretty much. Genetic defects, nutritional deficiencies, tissue damage, toxins, even infections. Think Vibrio cholerae, the bacteria that causes cholera. It works by messing with enzyme function.
And this link between enzyme activity and disease provides a powerful diagnostic tool then.
Precisely. Detecting and quantifying the activity of specific enzymes in blood, or other bodily fluids, or even cell extracts. It gives invaluable diagnostic information.
Like if your liver enzymes are high in a blood test.
Exactly. That can indicate liver damage because those enzymes have leaked out of the damaged cells.
So, okay, outside of keeping us alive and healthy day to day, where else might we find enzymes doing work?
Well, beyond their fundamental role in metabolism, enzymes turn up in some, well, surprising places. Rennin, for example, it's a protease.
Breaks down protein.
Right. It's essential in cheese production. It helps curdle the milk. And lactase, which many lactose-intolerant people rely on in supplement form.
Yeah.
It's used industrially to remove lactose from milk products.
I never thought about cheese making needing enzymes. What about other industrial applications?
Oh, definitely. Think about your laundry detergent. Proteases and amylases, the ones that break down proteins and starches, are often added to boost cleaning power against tough stains like, grass or gravy.
Makes sense.
And the pharmaceutical industry relies heavily on enzymes. For synthesizing complex drugs and antibiotics, enzymes can perform very specific chemical transformations, making sure the final molecule has the exact right structure to be effective. That stereospecificity is crucial.
Okay, so we know they're important, they're used everywhere. But what makes enzymes so exceptional at what they do? The text emphasizes their efficiency and specificity. Let's dive into that.
The efficiency is truly remarkable. Enzymes can enhance reaction rates by factors of, 10 to the 6th, up to 10 to the 9th, even more sometimes.
So millions to billions of times faster.
Exactly. Compared to the same reaction happening without a catalyst, it's like comparing a snail's pace to the speed of light, almost. And importantly, while some enzymes undergo temporary changes during the reaction, they always get regenerated back to their original state, ready for the next cycle.
And it's not just about speed, right?
Yeah.
It's also about precision. That's selectivity.
Absolutely. They're usually incredibly specific, often for just a single substrate molecule or maybe a small group of very closely related ones.
How do they manage that level of specificity?
It often comes down to the active site, the pocket, where the reaction happens. The idea of 3 points of attachment is often used to explain it. Think of it like needing 3 specific contact points to line up perfectly between the enzyme and the substrate.
Like a very specific handshake.
Sort of, yeah, a very precise fit. And because of this, isozymes can even distinguish between stereoisomers, molecules that are mirror images of each other.
You mean like left-handed and right-handed versions of a molecule? Chirality.
Exactly, chirality. Like your left and right hands, they look the same, but you can't superimpose them. Enzymes can often bind only one of those forms. For example, enzymes acting on sugars usually prefer the D form, while those acting on amino acids typically select the L form.
And they can even create specific chiral forms from non-chiral starting materials.
Yes. Lactate dehydrogenase is a classic example. It takes pyruvate, which isn't chiral, and converts it only to L-lactate, not a mixture of L and D.
So this incredible specificity.
Yeah.
What's the big picture consequence inside a living cell?
It means thousands of different chemical reactions can all be happening at the same time, in the same tiny space, completely independently, without interfering with each other.
Like a perfectly organized factory with thousands of specialized machines?
Precisely. It allows for simultaneous and independent control over all the diverse biochemical processes needed for life. It's biochemical order out of potential chaos.
Now, with so many enzymes doing so many different things, there must be a system for naming and organizing them. I know the text mentions the early days, things were a bit loose.
Right. Early on, enzymes were often named by just adding ase to something descriptive, maybe the substrate, like lactase for lactose, or the reaction type. We still use some of those old names, pepsin, trypsin, amylase. They're familiar.
But that probably got confusing as more enzymes were found.
Yeah.
Multiple names for the same thing, maybe.
Exactly. It became clear a more systematic approach was needed. That's where the International Union of Biochemistry, the IUB, stepped in.
Okay, the IUB system, what's the gist?
They developed a comprehensive system where every known enzyme gets a unique, unambiguous name and a code number, the EC number, for enzyme commission number. This system classifies enzymes into 6 main classes based purely on the type of reaction they catalyze.
6 main classes. Okay, let's run through them. What's the first one?
First are the oxidoreductases. As the name suggests, they handle oxidation reduction reactions, transferring electrons, essentially.
Got it. Electron movers, number 2.
Transphrases. These move functional groups like a phosphate group or a methyl group or a sugar group from one molecule to another.
Group swappers. Okay, third.
Hydrolysis. These break chemical bonds using water. Hydrolysis. Think digestion breaking down large molecules into smaller ones.
Right, water-based demolition, number 4.
Lyases. They also break bonds, often CC, CO, or CN bonds, but they do it without hydrolysis or oxidation. Often they form double bonds or rings in the process.
Bond breakers, but no water needed. 5th class.
Isomerases. These catalyze rearrangements within a single molecule. They change the geometry or structure, converting one isomer to another.
Molecular reshufflers. And the last one, number 6.
Ligases. These join 2 molecules together, think synthesis, building larger molecules. This process usually requires energy, often supplied by breaking down ATP.
Molecular construction crew powered by ATP. Okay. The text gives hexokinase as an example.
Okay.
EC 2.7.1.1. That number's complicated. Can you quickly decode what it tells a biochemist?
Sure. The 2 means it's a transferase, class 2. The 7 means it transfers a phosphorus-containing group. The 1, after that, means the acceptor of that group is an alcohol group. And the final one specifies which alcohol group, in this case, on the 6th carbon of a hexose sugar.
Wow, that's incredibly specific.
It is. The IUB names and numbers are precise, which is vital for clear communication in research. But, they can be mouthfuls.
Yeah, ADPD hexose 6 phosphotransferase versus hexokinase.
Exactly. So in everyday lab talk, we often still use the shorter common names like hexokinase, even if they're sometimes a bit less specific than the full EC name. It's a trade-off between precision and convenience.
Okay, so we have the enzymes themselves, usually proteins, But you mentioned they often don't work alone. They need helpers.
That's right. Many enzymes require additional chemical components called cofactors or coenzymes. These can be simple metal ions or more complex organic molecules.
What do these helpers actually do?
They participate directly in the catalysis. Sometimes they help bind the substrate. Sometimes they're directly involved in the chemical transformation. They essentially extend the catalytic capabilities beyond what the 20 standard amino acid side chains can offer.
The text makes a distinction between prosthetic groups and cofactors. What's the key difference there?
It mainly comes down to how tightly they're bound to the enzyme. Prosthetic groups are very tightly, sometimes even covalently, bound to the enzyme protein. They're essentially a permanent part of the enzyme structure.
Okay, like built-in tools. Examples.
Things like pyridoxal phosphate, PLP, flavin mononucleotide, FMN, flavin adenine dinucleotide, FAD, thiamine pyrophosphate, TPP, biotin, metal ions like iron, zinc, copper. Even the heme group in hemoglobin, though that's not an enzyme, it illustrates a prosthetic group.
Right. And cofactors, how are they different?
Cofactors bind much more loosely, reversibly, associating with the enzyme only transiently, usually during the reaction cycle. Often they actually associate with the substrate as much as the enzyme.
So they're more like temporary assistants that come and go.
Exactly. Metal ions are a very common type of cofactor. We sometimes talk about metal-activated enzymes where the ion binds weakly versus metalloenzymes where the metal is tightly bound like a prosthetic group. And it's estimated that maybe a third of all known enzymes require a metal ion for activity one way or another.
And then there are coenzymes. Are they different again?
Coenzymes are a subset of cofactors, specifically the more complex organic or metallo-organic molecules. They act more like transient carriers of specific functional groups. Think of them as shuttles.
Shuttles. Carrying what?
Carrying things like electrons like NAD+ and FAD, or acyl groups like coenzyme A, or single carbon units like derivatives of folate. They pick up a group from one reaction and deliver it to another. They act almost like co-substrates because they get chemically changed during the reaction. But unlike regular substrates, they get regenerated in a subsequent reaction, often by a different enzyme.
And many of these cofactors and coenzymes come from vitamins, right?
Absolutely. Many of the B vitamins are precursors to essential coenzymes. Niacin for NAD and NADP, riboflavin for FMN and FAD, pantothenic acid for coenzyme A, thiamine for TPP, folic acid, vitamin B12, the list goes on.
So that's a direct link between nutrition and basic cellular chemistry if you lack a vitamin.
The corresponding coenzyme can't be made, and the enzymes that depend on it won't function properly. That's the biochemical basis for vitamin deficiency diseases.
Fascinating. Okay, let's move to where the magic happens, the active site. The text mentions an early observation about substrates protecting enzymes from heat.
Yes, early biochemists noticed that if you heated an enzyme gently, it would lose activity denature. But if you added its substrate first, the enzyme was much more resistant to heat.
Which led Emil Fischer to propose
the enzyme-substrate complex, or ES complex. He reasoned that the enzyme and substrate must bind together to form a complex, and this complex was somehow more stable than the enzyme alone. This led to his famous lock and key analogy.
the idea that the substrate, the key, fits perfectly into a rigid active site, the lock.
Exactly. It was a brilliant insight for its time and explained the specificity very well.
So what is the active site like structurally?
It's typically a cleft or pocket on the surface of the enzyme, formed by specific amino acid residues brought together by the protein's folding. It's not just a passive binding dock, though.
It does more than just hold the substrate.
Oh, definitely. The active site is the site of catalysis. It binds the substrate and positions them perfectly relative to each other and to the catalytic amino acid residues, cofactors, and so on.
And it creates a special environment.
Yes, that's crucial. The active site often excludes bulk water, creating a microenvironment with a different polarity or hydrophobicity or pH than the surrounding cytoplasm. This can be critical for making the reaction happen or speeding it up.
Okay, so how do enzymes actually achieve these incredible rate enhancements? The text outlines 4 general strategies. First is catalysis by proximity. What's that about?
It's simply about bringing reactants together. For 2 molecules to react, they need to collide with sufficient energy and in the correct orientation. An enzyme's active site effectively binds the substrate molecules, holding them close together and in the perfect alignment for reaction.
So it increases the effective concentration and gets the geometry right.
Precisely. Just getting them close and oriented correctly can provide a significant rate boost, maybe 1,000-fold or more.
Second strategy, acid-base catalysis.
Here, amino acid side chains within the active site, or sometimes prosthetic groups, act as proton donors, acids, or proton acceptors, bases. Many organic reactions involve proton transfers, and having these groups perfectly positioned can stabilize transition states or make reactants more reactive.
And the text mentions specific versus general acid-base catalysis.
Right. Specific acid-base catalysis involves only protons, H+, or hydroxide ions, OH, from water. The rate depends only on pH. General acid-base catalysis involves proton transfers to or from other molecules like those enzyme side chains, and the rate depends on the concentration of those specific acids or bases. Enzymes typically use general acid-base catalysis.
Okay, third strategy, catalysis by strain. This sounds like the enzyme is, well, bending the substrate?
In a way, yes. When an enzyme binds its substrate, it sometimes forces the substrate into a conformation that's electronically or physically strained, resembling the transition state of the reaction.
The transition state being that high-energy, unstable intermediate point between reactant and product.
Exactly. By binding the substrate in a way that mimics this transition state, or by binding the transition state itself even more tightly than the substrate, the enzyme lowers the activation energy needed to reach it. Linus Pauling was a key proponent of this idea, transition state stabilization.
And this has practical applications in drug design.
Absolutely. If you know what the transition state looks like, you can design molecules called transition state analogs that mimic it very closely. These often bind extremely tightly to the enzyme's active site and act as potent inhibitors. Many drugs work this way.
Very clever. And the fourth strategy, covalent catalysis.
This involves the enzyme forming a temporary covalent bond with the substrate as part of the reaction pathway. This creates a reactive enzyme-substrate intermediate.
So the enzyme itself becomes chemically modified for a short time.
Yes, but it's always regenerated back to its original state by the end of the reaction cycle. This strategy often provides a new reaction pathway with a lower activation energy. It's common in reactions involving group transfers, and amino acids like serine, cysteine, or sometimes histidine are often involved in forming that covalent bond.
Now, Fisher's lock and key model was a great start, but it implied rigidity. The text introduces Koshland's induced fit model. How did that refine the picture?
Koshland recognized that enzymes aren't static, rigid locks. He proposed the induced fit model, suggesting that the binding of the substrate actually induces a change in the conformation, the shape, of the enzyme's active site.
So the lock changes shape slightly as the key goes in.
Exactly. It's often compared to a hand fitting into a glove. The glove enzyme molds itself around the hand substrate for better fit. This conformational change can help position the catalytic groups correctly, strain the substrate, or exclude water.
And the substrate changes shape, too.
Yes, it's a reciprocal process. The enzyme induces changes in the substrate, using the energy released from binding to help distort the substrate towards the transition state. It's a much more dynamic picture than the simple lock and key.
And we have evidence for this dynamic behavior.
Oh, yes. Biophysical studies that can track the movements of enzymes as they bind substrates clearly show these conformational changes happening. It's now a widely accepted refinement of the original model.
So, summarizing the models, lock and key is a rigid fit, while induced fit involves both enzyme and substrate changing shape for optimal interaction and catalysis.
That's a good way to put it.
Okay, let's look at some concrete examples. The text mentions HIV protease using acid-base catalysis. How does that work?
Right. HIV protease is an aspartic protease, like pepsin in our stomachs. It cuts polypeptide chains, which is essential for the HIV virus to mature. Its active site has two crucial aspartate residues.
And they act as the acid and base.
Yes. As shown in Figure 7-6, one aspartate acts as a general base. It grabs a proton from a water molecule, activating the water to attack the peptide bond. The other aspartate acts as a general acid, donating a proton to stabilize the leaving group as the bond breaks. They work in concert.
So those two aspartates are perfectly positioned to manipulate that water molecule and break the bond.
Precisely. And the environment around them helps ensure one is protonated, acting as acid, and the other is deprotonated, acting as base at the start.
Next example. Chymotrypsin, using covalent catalysis. This involves that catalytic triad.
Yes. Chymotrypsin is a serine protease. It uses a catalytic triad of 3 amino acids, serine 195, histidine 57, and aspartate 102, using the numbering from the bovine enzyme.
What does the triad do?
It acts like a charge relay network or a proton shuttle. The aspartate helps orient the histidine, and the histidine acts as a base, pulling a proton off the serine 195 hydroxyl group. This makes the serine oxygen a very strong nucleophile.
A nucleophile wants to attack electron-deficient centers.
Exactly. This superreactive serine oxygen attacks the carbonyl carbon of the peptide bond in the substrate. This forms a temporary covalent bond, the acyl enzyme intermediate, and breaks the peptide bond, releasing the first part of the substrate.
Okay, so half the job is done, but the enzyme is stuck to the other half.
Right. Then a water molecule comes in. The histidine, again helped by the aspartate, acts as a base, activating the water to attack the acyl enzyme intermediate. This breaks the covalent bond between the enzyme and the remaining part of the substrate, releasing the second product and regenerating the original enzyme with its serine hydroxyl group intact.
That's quite a complex dance. Figure 7-7 really helps visualize that proton shuffling.
It is intricate but very efficient. Trypsin and elastase-related serine proteases use a very similar mechanism, just with different substrate specificities determined by the shape of their binding pockets.
And the third example, fructose 2-6 bisphosphatase, also uses covalent catalysis, but with a different triad.
Yes, this enzyme removes a phosphate group. Its mechanism, shown in Figure 7-8, involves a catalytic triad of 1 glutamate and 2 histidine residues. One of the histidines actually forms a temporary covalent phosphohistidyl intermediate during the reaction.
So similar strategy, forming a covalent intermediate, but using different amino acid players.
Exactly. And other residues in the active site, like lysine and arginine, help bind the substrate and stabilize charges during the reaction.
It's striking how these families of enzymes, like serine proteases, use the same core mechanism but target different molecules. How did that evolutionary divergence happen?
It often starts with gene duplication. A gene encoding an enzyme gets copied. Then the two copies can evolve independently. One might retain the original function, while the other accumulates mutations that change its substrate binding pocket, allowing it to act on a new substrate while still using the same basic catalytic machinery.
So chymotrypsin and trypsin are examples of these divergent homologs. Same family, different targets.
Exactly. They are homologous proteins sharing a common ancestor. And when you compare their sequences, you find that the key catalytic residues, the serine, histidine, and aspartate of the triad, are conserved, meaning they appear in the same relative positions.
Because they're essential for the mechanism.
Right. These evolutionarily conserved residues tell you they're critically important for function.
And what about isozymes? The text mentions them too. Are they the same as homologs?
Not quite. Isozymes, or isoenzymes, are different forms of an enzyme within the same organism that catalyze the same reaction. They often arise from gene duplication, too, or sometimes from alternative splicing of RNA from a single gene.
But if they catalyze the same reaction, what's the point of having different versions?
They usually differ in other properties, like their regulatory characteristics, their kinetic parameters, like how tightly they bind substrate, or their location within the cell or specific tissues. This allows for fine-tuning of the same reaction under different conditions or in different parts of the body. For example, different isozymes of lactate dehydrogenase are found in heart versus skeletal muscle, reflecting the different metabolic needs of those tissues.
So isozymes provide tissue-specific regulation or adaptation rather than different substrate specificity.
Got it. Now, you mentioned earlier that enzymes are hard to detect directly because they're in low concentrations, but their activity amplifies their presence.
Yes, that catalytic power is key. Because one enzyme molecule can process thousands or millions of substrate molecules per second, we can easily measure the rate of product formation or substrate disappearance. Under controlled conditions, this rate is proportional to the enzyme concentration.
So enzyme assays are standard practice in research and clinical labs. Absolutely essential, and the technology keeps improving. We now have single molecule enzymology.
Observing just one enzyme molecule at work.
Exactly. Using advanced microscopy and detection techniques, scientists can watch individual enzyme molecules catalyze reactions one event at a time. This gives incredibly detailed information about the mechanism and dynamics that you'd miss just looking at the average behavior of billions of molecules in a test tube.
And this sensitivity is also leveraged in drug discovery, high-throughput screening.
Yes. Since many drugs target enzymes, pharmaceutical companies need to screen huge libraries of potential drug compounds, sometimes millions, to see if any inhibit a target enzyme. High-throughput screening, HTS, uses robotics, microfluidics, and rapid detection methods to automate these enzyme assays on a massive scale.
Finding the needle in the haystack.
Pretty much. It works hand in hand with combinatorial chemistry, which generates those vast libraries of compounds to test.
And ELISA's enzyme-linked immunosorbent assays use enzyme to detect other things?
Right. ELISAs are incredibly sensitive tests used to detect the presence of specific proteins, often antibodies or antigens, not necessarily enzymes themselves. They work by linking an antibody that recognizes the target protein to a reporter enzyme.
Like alkaline phosphatase or horseradish
Those are
peroxidase?
common ones, yes. If the target protein is present in the sample, say blood serum, the antibody enzyme conjugate binds to it. Then you add a substrate for the reporter enzyme that produces a colored or fluorescent product. The amount of color or fluorescence tells you how much of the target protein was there. The enzyme just acts as an amplification system to make the detection highly sensitive.
Very clever.
Yeah.
Okay, let's get practical. How are these assays actually done? The text mentions spectrophotometric assays for dehydrogenases.
Right. Many dehydrogenases use the coenzymes NAD plus or NADP plus tira. The key thing is that the reduced forms, NADH and NADPH, absorb light strongly at a wavelength of 340 nanometers, while the oxidized forms, NAD plus and NADP plus era, do not.
So you can just watch the absorbance change in a spectrophotometer.
Exactly. If the reaction produces NADH or NADPH, the absorbance at 340 nanometers goes up over time. If the reaction consumes them, it goes down. The rate of change in absorbance is directly proportional to the enzyme's activity. It's a very convenient and common assay method.
But what if your enzyme's reaction doesn't involve NADPH or any other convenient color change?
That's common. One trick is to use an artificial or synthetic substrate. You design a molecule that the enzyme will act on, but when it does, it releases a product that is colored or fluorescent. A classic example is using p-nitrophenyl phosphate, PNPP, to assay phosphatases. PNPP is colorless, but when the phosphate is removed, it releases p-nitrophenolate, which is bright yellow.
So you create a detectable signal. What about coupled assays?
Couple assays are another powerful technique, especially when the enzyme you're interested in, let's call it enzyme 1, doesn't have an easy assay itself. You link its reaction to a second reaction catalyzed by an auxiliary enzyme, enzyme 2, which does produce a detectable signal, often involving NADPH.
How does that work?
The product of the reaction catalyzed by enzyme 1 becomes the substrate for enzyme 2. You add both enzymes to the reaction mix, along with the substrate for enzyme 1, and any necessary cofactors for enzyme 2, like NAD+. You make sure you have plenty of enzyme 2, so it's not the limiting factor.
So enzyme 2 immediately converts the product of enzyme 1.
Right, as soon as enzyme 1 makes its product, enzyme 2 acts on it, producing NADH or consuming it. You then monitor the change in absorbance at 340 nanometer. Because enzyme 2 is working so fast, the rate of change you see actually reflects the rate of enzyme 1.
So you're indirectly measuring the first enzyme's activity through the second one, like a relay race where you only time the second runner, but they start immediately when the first finishes.
That's a good analogy. Figure 711 shows an example coupling hexokinase activity to glucose 6-phosphate dehydrogenase, monitoring NADPH production.
It's clear how vital these assays are. Let's talk more about their use in clinical diagnosis. You mentioned leaked enzymes indicating tissue damage.
Yes, analyzing enzymes in blood plasma or serum is a cornerstone of clinical chemistry. While some enzymes are normally present and functional in blood, like those involved in clotting or lipoprotein lipase, many others are typically found inside cells.
So if they show up in the blood in high amounts.
It usually means cells containing that enzyme have been damaged or died, releasing their contents into the bloodstream. Measuring the levels of specific enzymes known to be abundant in particular tissues like liver, heart, pancreas, or muscle can help pinpoint the site of injury.
And the timing matters too.
Absolutely. Different enzymes leak out at different rates. Cytoplasmic enzymes usually appear in the blood faster after injury than enzymes located inside organelles like mitochondria. Following the rise and fall of different enzyme levels over time can help determine when the injury occurred and how it's progressing. Table 7.1 in the source lists several clinically important enzymes.
Like ALT and AST for liver, amylase and lipase for pancreas, creatine kinase for muscle.
Right. But it's important to remember that few enzymes are absolutely tissue-specific. ALT is high in liver, but also present elsewhere. So results always need interpretation in the context of the patient's history, symptoms, and other tests. Sensitivity and specificity are key considerations.
The text uses myocardial infarction, MI, a heart attack, as a detailed example. How did enzyme analysis help there historically?
Diagnosing an MI quickly is crucial for starting treatment. Historically, doctors looked at enzymes like aspartate aminotransferase, AST, and alanine aminotransferase, ALT, which rise after heart muscle damage, but they're not very specific to the heart. Lactate dehydrogenase, LDH, was also used.
LDH has isozymes, right? Different forms in different tissues.
Yes, LDH has 5 main isozymes separable by electrophoresis. The pattern of LDH isozymes in the blood, shown in Figure 712, could give clues about heart damage, specifically LDH1 and LDH2 increasing relative to others. But LDH levels rise relatively slowly after an MI.
So not ideal for rapid diagnosis. What came next?
Creatine kinase, CK, also called creatine phosphokinase, CPK. CK also has isozymes. CKMM is predominant in skeletal muscle, CKBB in the brain, and CKMB is found in heart muscle, though also some in skeletal muscle. Measuring CKMB levels provided a more specific and earlier indicator of heart damage than total CK or LDH. For a while, CKMB was the gold standard.
But now it's mainly used for skeletal muscle disorders, like muscular dystrophy.
Primarily, yes, because an even better marker for heart attacks emerged.
Which is?
Plasma troponin. Troponin is a complex of 3 proteins involved in muscle contraction. There are cardiac-specific versions of 2 of these proteins, troponin I, CTNI, and troponin T, CTNT.
And these are only found in heart muscle?
Essentially, yes. They aren't found in smooth muscle, and the cardiac forms are distinct from the skeletal muscle forms. When heart muscle cells are damaged, CTNI and CTNT leak into the blood. Levels start to rise within 2 to 6 hours after an MI, peak within 12-24 hours, and can stay elevated for days.
So a longer diagnostic window and higher specificity.
Exactly. Immunological tests that specifically measure cardiac troponin I or T are now the preferred biomarkers for diagnosing myocardial infarction. They are very sensitive and specific indicators of heart muscle injury.
Beyond diagnosing injury, what are some other clinical uses of enzymes?
Well, as we mentioned, enzymes are used in lab tests to measure other things, like glucose oxidase being used to measure blood glucose levels. And enzymes themselves are used as therapeutics.
Drugs that are actually enzymes.
Yes. For example, tissue plasminogen activator, TPA, or streptokinase, are enzymes used to dissolve blood clots during a heart attack or stroke. Trypsin can be inhaled to help clear thick mucus in cystic fibrosis patients. And for certain genetic lysosomal storage diseases where patients lack a specific enzyme, intravenous infusion of a recombinant version of the missing enzyme can be life-saving therapy. Examples include Gaucher disease, Pompe disease, Fabry disease, several others listed in the text.
Incredible. Enzymes are also used in diagnosing genetic and infectious diseases, particularly using DNA-modifying enzymes.
Right. This leverages the specificity of enzymes that act on DNA. Restriction endonucleases or restriction enzymes are a key example.
These are the enzymes that cut DNA only at specific recognition sequences.
Precisely. They act like molecular scissors, cutting DNA wherever they find their specific target sequence, which is usually 4 to 6 or more base pairs long.
How's that used diagnostically? RFLPs?
Restriction fragment length polymorphisms, or RFLPs. The idea is that if a mutation occurs within a restriction enzyme's recognition site, the enzyme won't cut there anymore. Or a mutation might create a new site. So if you take DNA from different individuals, cut it with the same restriction enzyme, and separate the fragments by size using gel electrophoresis, you might see different patterns of fragments if they have variations in those restriction sites.
And this can reveal genetic differences linked to disease.
Yes. Historically, RFLP analysis was used for prenatal diagnosis of diseases like sickle cell anemia or Huntington's disease, and also in forensic DNA fingerprinting. The limitation was needing a relatively large amount of DNA.
Which brings us to PCR.
Exactly. The polymerase chain reaction, or PCR, has largely replaced RFLP for many applications because it's vastly more sensitive. PCR uses a heat-stable DNA polymerase enzyme, like Taq polymerase, and short DNA primers to make millions or billions of copies of a specific target DNA sequence, even starting from just a tiny amount.
So you amplify the region of interest first.
Right. Then you can analyze that amplified DNA, perhaps sequence it directly, or test it with restriction enzymes. PCR is used everywhere now, detecting genetic mutations, identifying pathogens like bacteria or viruses. By amplifying their specific DNA forensics research, it's revolutionary.
To really study enzyme structure and function in detail, you need pure enzyme. How does recombinant DNA technology help with that? It must be hard to isolate one protein from thousands in a cell.
It used to be incredibly difficult, but now, using recombinant DNA techniques, we can clone the gene that codes for the enzyme we're interested in. Then we can insert that gene into a host organism, usually bacteria like E. coli or sometimes yeast, and trick the host cells into producing large quantities of our desired enzyme protein.
So you turn bacteria into enzyme factories.
Essentially, yes. There can be limitations. Sometimes bacterial cells don't fold complex animal proteins correctly or perform necessary post-translational modifications. In those cases, researchers might use cultured animal cells or insect cells, using baculovirus vectors, for expression, though these systems are often more complex and expensive.
Once you've made the protein, you still need to purify it. The text mentions affinity chromatography using tags.
Affinity tags are a very common and powerful purification strategy. You modify the cloned gene so that the protein produced has a short extra sequence, a tag, attached to one end, usually the N or C terminus. This tag is designed to bind very specifically to a particular substance immobilized on a chromatography column.
Like the his tag.
The polyhistidine tag, or his tag, usually 6 histidine residues in a row, is very popular. His tags bind strongly to immobilized divalent metal ions like nickel, Na2+, or cobalt, CO2+. You pass the crude cell extract over a column containing these immobilized metal ions. The his-tagged protein sticks, while most other cellular proteins wash through. Then you elute your tagged protein by adding a high concentration of imidazole, which competes for binding to the metal ions.
And the GST tag.
Another common one is the glutathione S transferase, GST tag. GST is a protein itself that binds specifically to glutathione, so you use a column with immobilized glutathione to capture the GST-tagged fusion protein.
And you can often remove the tag afterwards.
Yes. Often the tag sequence is designed to include a specific cleavage site recognized by a highly specific protease like thrombin or TV protease. After purification, you can treat the protein with this protease to cut off the tag, leaving you with just your target enzyme.
So recombinant DNA gives us the protein. How do we use it to understand how the enzyme works? Site-directed mutagenesis.
Site-directed mutagenesis is a fantastic tool for probing enzyme mechanisms. Once you have the clone gene, you can make precise changes to its DNA sequence, altering the codon for a specific amino acid.
So you can swap out, say, an aspartate you think acts as a base catalyst for an alanine, which can't.
Exactly. You express this mutated version of the enzyme, purify it, and then test its activity. If the activity is lost or dramatically reduced, it strongly suggests that the residue you changed was indeed important for catalysis. Combining this with kinetic studies and structural analysis, like X-ray crystallography, of both the original and mutated enzymes, allows researchers to pinpoint the roles of specific amino acids in substrate binding and the chemical steps of catalysis.
Okay, finally, let's talk about a major curveball in enzymology.
Yeah.
Ribozymes, the idea that not all enzymes are proteins.
This was a truly paradigm-shifting discovery. For decades, it was dogma that biological catalysis was the exclusive domain of proteins. But in the early 1980s, Thomas Seck and Sidney Altman, who shared the Nobel Prize for this, independently discovered that RNA molecules could also act as catalysts.
Seck was studying RNA processing in tetraamina.
Yes, he found that the splicing of a ribosomal RNA molecule, cutting out an intron and joining the exons, could happen in a test tube with no protein present. The RNA molecule itself was catalyzing the reaction. He called this catalytic RNA a ribozyme.
And other ribozymes have been found since.
Yes, quite a few, although still far fewer than protein enzymes. Most known natural ribozymes catalyze reactions involving RNA itself, like cleavage or splicing. Examples include the hammerhead ribozyme, the hepatitis delta virus ribozyme, and the self-splicing group 1 and group 2 introns.
And the biggest ribozyme of all is the ribosome itself.
That's the current understanding, and it's mind-blowing. The ribosome responsible for making all proteins is this huge complex of ribosomal RNA, rRNA, and proteins. For years, people thought the RNA was just a scaffold and the proteins did the catalysis.
That turned out to be wrong.
Completely backwards, actually. Landmark studies, particularly by Harry Noller, Adi Yonath, Venki Ramakrishnan, and Thomas Stites, who also shared a Nobel Prize, showed that the active site for peptide bond formation resides entirely within the RNA. The ribosomal proteins are mostly on the periphery, stabilizing the structure, but the chemistry is done by RNA. The ribosome is fundamentally a ribozyme.
Wow, and this discovery had huge implications for thinking about the origin of life. The RNA World hypothesis.
Profound implications. Before ribozymes, the origin of life posed a chicken and egg problem. DNA holds the information to make proteins, enzymes, but you need enzymes to replicate DNA and make proteins. Which came first?
Right.
The discovery that RNA could both store information, like DNA, and catalyze reactions, like proteins, offered a potential solution. The RNA World hypothesis suggests that early life may have been based primarily on RNA. RNA molecules could have served as both the genetic material and the main catalysts before DNA took over as the more stable information store and proteins took over as the more versatile catalysts.
So DNA and proteins evolved later.
That's the idea. DNA is chemically more stable than RNA for long-term information storage. And proteins, with their 20 different amino acid building blocks, offer a much wider range of chemical functionalities and structural possibilities for catalysis than RNAs for bases. The ribosome might even be seen as a relic from this transitional period, an RNA machine that learned to make proteins.
So why do we still have ribozymes today? Why didn't proteins take over all catalysis?
Good question. Perhaps for some specific reactions, particularly those involving RNA itself, RNA catalysis remains sufficiently efficient, or evolving a new protein catalyst for those tasks was just never selected for. The ribosome itself is so central and complex, maybe replacing its core catalytic RNA with protein was just too difficult or disruptive evolutionarily.
Wow, this has been an incredible deep dive into the world of enzymes, stretching from basic definitions all the way to the origins of life.
Indeed. To maybe recap some key takeaways.
Yeah.
Enzymes are these highly specific, incredibly efficient biological catalysts, absolutely essential for life.
Right. They use a whole toolkit of strategies, proximity, acid-base chemistry, strain, covalent bonds, often needing helpers like cofactors and coenzymes derived from vitamins.
And their activity isn't just vital inside cells. It's a powerful tool we use constantly for diagnosis, for therapy, in industry, and in fundamental research.
And the discovery of ribozymes really opened up our view, showing catalysis isn't just for proteins and giving us hints about early life.
Absolutely. It's a field that connects chemistry, biology, medicine, and even evolution.
It really makes you think. Are there other kinds of catalytic molecules hiding in plain sight within biological systems? What other surprises might be waiting?
That's the exciting part of science, isn't it? There's always more to discover. This deep dive has really only scratched the surface. You could spend a lifetime exploring just one enzyme's mechanism, or mapping out metabolic pathways, or delving into the rapidly expanding world of RNA biology.
Definitely plenty more to explore. We encourage you to dig deeper into any area that caught your interest today.
Keep asking questions.
Thanks for joining us on this deep dive into the essential world of enzymes. Until next time, keep exploring the incredible complexity of life around us.